Mitosis culminates usually with the physical partitioning of two daughter cells from each other, and we call that phase in which the cell actually divides cytokinesis. Cytokinesis in animal cells is special, and I'm going to begin our discussion of it by describing some phenomenological rules observed by experiment. Much of those experiments performed on sea urchin eggs, as it happens, sand dollar eggs, things like that. So, number one, let's consider an egg with a mitotic spindle in it. This first rule is going to come as a surprise really to nobody. Animal cell cytokinesis is rigged up such that the cleavage plane always bisects the spindle. Even if the spindle is offset, eccentrically positioned somehow. So let's say we have a spindle all the way over here on one side of the cell. Meiosis represents an extreme case of eccentric spindle position, but unequal cell divisions are common, especially during development. The rule would say that the cleavage furrow should divide the cell like that, in this case, into two very different sized daughters, right? Now, one of the practical ways to interpret this is if you were the designer of a developmental program and you wanted to choreograph some asymmetric cell division, what you would have to do is rig up something that would drag the mitotic apparatus over toward the side that you wanted to form a small cell on. This can be done experimentally as well. So let's say we come upon this cell with a blunt probe, and I squish it. I made a living for 2 decades or more doing this kind of thing to cells, by the way. What I've done here is displace one of the spindle poles so that it's no longer, so that the mitotic apparatus is no longer centered. And as a consequence, the cleavage furrow that might have bisected the cell this way will now do so this way. So that's rule number one. Number two, more or less as a consequence of experiments like these, we can show that the entire surface of an animal cell is usually competent to mount whatever it is that produces the cleavage furrow. An inference from that is that there's no pre-pattern. The cell doesn't know where it's going to divide until it comes time to do so. Or if there is a pre-pattern, it's one that can be overruled, as in this experiment. Third rule also sort of falls along these same lines. Let's consider a cell that has already commenced to divide. It's a little uneven. Oh well. Now I'm going to bring the probe over here and insert it that way into the pole of this dividing cell such that I shove the mitotic apparatus out from under the cleavage furrow that's already started to make progress through the cell. So that's going to do something like this. Creating a mismatch between the original position of the cleavage furrow and the new spindle midplane. In order to partition the centrosomes and the segregated chromosomes into the two daughter cells, the cell better correct this induced mismatch. So this cleavage furrow will regress, and the cell initiates a new one over the spindle midplane. This is the position of the original cleavage furrow, roughly, and the new cleavage furrow is induced, again, across the spindle midplane. Now, if I'm the spiteful sort and happen to have 2 micromanipulators handy, I might decide, well, let's see how long I can keep this up. And I'll come along to the other side with a blunt probe. Okay, well, I'm just trying to fix that up so my drawing is slightly more accurate there. So if I displace the mitotic apparatus again, I will once again have a mismatch between the spindle position. This cleavage furrow in progress will regress, and a new one will be induced over the new position of the spindle midplane. With a sand dollar egg or a starfish egg, you can keep doing this kind of thing, pushing it back and forth for a pretty long window of time, basically as long as the spindle remains coherent enough to push around, demonstrating that the cell cortex is continually responsive to some sort of cue coming from the mitotic apparatus whose geometry is communicating to the surface where it is. However, instead of a blunt probe this time, I'm going to come upon a cell that has initiated cleavage with a sharp needle that's big enough to actually remove some cytoplasm, and I'm going to suck the spindles right out of that cell. Now, I should mention, you can also do this with drugs that poison microtubule assembly. It's been done in a variety of different ways on a variety of different cell types. What do you think is going to happen? Now, rule number 3 was that the cleavage furrow continually tracks the mitotic apparatus. When we take away the mitotic apparatus, the cleavage furrow nevertheless continues. That is, it's sensitive to the position of the mitotic. It's sensitive to the position of the mitotic apparatus, but once established, the cleavage furrow continues even in the absence of any microtubules, chromosomes, or whatever else is in there. Number 5 is a fairly straightforward one. The cell surface is unresponsive before the metaphase-anaphase transition. In the cell cycle. The cell cortex will not respond to a metaphase spindle, no matter how close you shove it to the cell surface with a needle or whatever. As soon as chromosomes segregate, as soon as they're scissored apart in anaphase, the cortex begins the process of producing whatever the cytokinetic apparatus is, and the cleavage furrow commences. And that continues for some time. Now, in the normal course of events, when somebody isn't shoving the spindle back and forth with a needle, cleavage in something like a sand dollar egg might take 10 minutes, as it happens, there's about a 25-minute or half-hour window where the cell could continue responding to spindle position. And that can be experimentally demonstrated by such physical manipulations. Number 6. Now I'm going to come along to this egg, and either by inserting a needle, or you can actually also do it by squishing, but it's better to do it with a needle inserted into the cytoplasm so that you don't otherwise influence the geometry of the cell surface. So let's say I've stuck a needle in there and moved it so as to slide the spindle up closer to one side. The cleavage furrow initiates on the side closest to the spindle. Another way to say that is, the closer the spindle is to the surface, the sooner that piece of surface will initiate the contractility that causes the cleavage furrow to ingress. Consequently, the progression of cell division might look like this in such a cell. With one side initiating furrow ingression long after the other and leading to this highly asymmetric progression. Now, this kind of experiment is not only done by mean guys with needles, it is done by nature as well. In particular, in the eggs of most jellyfish and other cnidarians, the mitotic apparatus is displaced all the way up here towards the animal pole. It's a relatively small mitotic apparatus, and it just forms up there. It happens to be the site where meiosis took place. The sperm happens to enter near the site of meiosis in many cnidarians, and they produce their first cleavage furrow, and all of the subsequent furrows of embryonic development in an extremely asymmetric way, referred to as unilateral furrowing for obvious reasons. This happens to be a trait shared by some other cells, and even sand dollars and starfish to a certain extent. While the mitotic apparatus is never exactly in the middle of the cell. Okay, I'm going to pause there and say, astonishingly, these 6 rules I've articulated, almost none of these apply to other eukaryotic cells. So for example, land plants, they do have a pre-pattern. You can see it as a band of microtubules that forms before prophase in a cell that's about to undergo mitosis. They know where they're going to divide. It's up to the spindle to find its way there. Later on, we'll see that's true of diatoms as well. Of course, in many eukaryotes, the exact rules for cytokinesis are not clearly known, but in the case of land plants, just to contrast one particularly well understood example, cytokinesis doesn't even involve the ingression of a furrow like this. In land plants, the mitotic apparatus collects vesicles at the midplane where the chromosomes used to be, and those vesicles fuse. This is the remains of the mitotic apparatus. Ignoring everything about where the cell surface is, these vesicles fuse in the center here. This so-called phragmoplast mechanism builds a cell plate, which in the case of land plants includes cellulose to make a new cell wall, partitioning the cytoplasm effectively from inside out. Totally different mechanism of cytokinesis. And it is of interest to inquire, wow, how come the mechanisms of cytokinesis might be so different from one eukaryotic group to another when the mechanisms for things like ciliary motility or cell cycle control are so strongly conserved. Next experiment also involves needles. In this case, I'm going to skewer the cell down on one side of the cleavage furrow as it initiates with a big stiff rod. So sticking a great big stiff object in this side, and then up here on this side of the hoop, we'll put in a slender little needle. So this one's flexible, and this one's stiff. And in fact, it has to be stiff enough to restrain progress by the cleavage furrow. What happens here is that the ingression of the furrow bends the flexible needle, demonstrating what exactly? What's the point of this? Just more tricks with unfortunate sand dollar eggs. Well, two things. First of all, actually, second of all, if you calibrate the stiffness of this of the flexible needle, if you measure how much force it takes to bend that needle by a certain amount, then by doing this experiment, you could measure how much force the contractile apparatus deforming the cell surface actually exerts. But first, more important, this demonstrates that the cleavage furrow actually involves a constriction of the cell surface. What else could be happening, really? Well, what else could be happening is perhaps this furrow is deepening by the secretion of vesicles into this neighborhood. Maybe there's a bunch of little bubbles adding to the cell membrane, and that's how it makes progress through the cell. Kind of like the land plant mechanism working from outside in instead of from inside out, right? Now, that actually may happen to a certain extent, and indeed, vesicle formation is thought to complete cytokinesis in animal cells at the final event called abscission. But this experiment here, in which an inserted flexible barrier is actually bent, moved out of the way by the ingressing furrow, demonstrates that some continuous thing is in there and causes this constriction. So what is in there? When people finally got a look at dividing cells in electron microscopes with good enough fixatives to preserve microfilaments, what they saw is that in a metaphase cell, there's basically nothing much to speak of at the cortex, maybe a few actin filaments, not much there. By anaphase, just as the cell starts to change shape, and I'm trying to draw it having elongated a little bit in anaphase. Let's give it some longer astromicrotubules to represent anaphase. There's a narrow, slender band of actin filaments in just the part of the cell surface where the cleavage furrow is about to ingress. You can barely see this before furrowing is overtly taking place in telophase. So in telophase, recall the nuclei are reforming, the microtubules of these asters interacting with the cortex are actually being bent by the deformation of the cell surface during cytokinesis. You find this narrow belt, perhaps 10 microns wide, on the cortex where actin filaments are enriched, and by careful electron microscopy, one can see that those actin filaments are aligned along the hoop, along the circumference of the hoop of constriction. And those persist, that belt persists until the very end, when the furrow is completely necked down. The cells are already thinking about their next division, perhaps initiating DNA replication. There might be a little remnant of the spindle here. This band of actin, which also includes myosin, persists until the very end, and then it disappears. What finally accomplishes abscission, the final act here, is something else, something related to the secretory machinery. Okay, so metaphase. Now the appearance of actin filaments there, and also myosin, the fact that they're aligned along the hoop suggests directly an analogy to other contractile processes, muscle in particular, which was already well understood at the time people discovered what's in the cleavage furrow. Now, I want to point out slight irony. If you think about this hoop of actin filaments as something that's potentially generating the tension to constrict the cell surface, it was actually first discovered in the eggs of jellyfish, where there isn't a complete hoop. So if you imagine it working something like the elastic band in your underwear, it's not easy to see how it's going to work if it's just an arc instead of a hoop. The other place it was first observed, equally ironically, is in squid eggs. Squid eggs are giant. The cleavage furrow certainly doesn't bisect the whole cell. It's just a little crisscrosses on top of this giant cell where the nuclei inhabit an island of cytoplasm. Ultimately, they carve cells out of that. So, funny thing that this so-called contractile ring was first observed in cells where it couldn't possibly be a ring. Nevertheless, this notion of circumferential tension generation has dominated thinking about cytokinesis ever since. Let's take a moment to think about how it could work. Let's just imagine taking the hoop out of there at some intermediate point. I should make that hoop sort of saddle shape to match the shape of the cell surface. Could it really be analogous to a muscle? Might there be regular sort of contractile units in there with actin filaments sticking out of them, as in muscle, and myosin motors engaging these anti-parallel filaments to shorten these contractile units and thus drive constriction? That seems very unlikely for the following reason. Muscle has a rest length and a maximum contraction extent that they aren't very different from each other. So perhaps this could constrict optimistically like that before all of these units in there reach their maximum contraction length. You can't keep doing this forever. Now, of course, there might be clever ways to arrange these things, but what people see in the cleavage furrow is a band of parallel actin filaments. And it's important to note that the cleavage furrow starts out with the circumference of the entire cell, right? The entire equatorial circumference, and gets necked down to nothing. And that suggests, instead of behaving like a muscle, whatever this contractile apparatus is in here, it's taking itself apart as it goes. If it weren't, you'd see this band getting broader and broader as it constricts, right? That's what an elastic band would do. So let's say probably not for this mechanism, not muscle-like. What else can we imagine? Let's imagine instead that out of these actin filaments, okay, we know they run roughly parallel to each other. Let's make a kind of a spool. And of course, as you all know, if you take a spool of thread and you pull on the ends, you can wind that spool tighter, right? Well, you could certainly keep that going indefinitely as long as you have some sort of shrinking cylinder around which to wrap it, right? And you can imagine how perhaps you could mix the polarities of actin filaments so that myosin motors could engage with them. Maybe somehow this spool sort of walks along in adjacent threads or something like that. Problem is, it would take an awful lot of architecture. It's difficult to imagine how you'd make it responsive on a realistic timescale for cell division. And furthermore, you would probably see, as a consequence of this, you would probably see a great deal of twisting between the two nascent daughter cells as they divide. Now, in some cells, there is a little bit of a twist, but I mean, it goes like, not like spinning the one daughter around relative to the other. So probably not this either. Instead, the going idea at this point, the best idea that we can offer is something like the following. Somewhere on the cell surface, and we'll just imagine a segment of this equatorial surface here. Somewhere on the cell surface where the cleavage furrow is induced, some kind of node is induced to start spitting out actin filaments. They're going to grow out that way. So let's imagine that these nodes spin out actin filaments. They're assembling at the node, which means their pointed ends are out here. I'm going to adopt the convention now saying these are the minus ends and the plus ends, the growing ends are in here, the barbed ends. All these nodes, neighboring nodes, therefore encounter or emit toward each other filaments of opposite polarity. And look, here's two filaments where there's a barbed end over here and a barbed end over here. Imagine recruiting myosin motors that could get in there, latch onto these two filaments, and they could pull so that these nodes experience a force, bringing them closer together. But further imagine, okay, so let's put some more myosins in there that can do that. Further imagine that these nodes have a brief lifetime, that they're, as soon as they form, they spit out a bunch of actin filaments that might reach out to neighboring nodes. They might engage with myosins transiently, but then after a little bit of, after a short lifetime and a little bit of local contraction, they might fall apart and new nodes might form, spit out some new actin filaments and so on. So the idea is that a dynamic contractile network like this underlies the continual progression of the cleavage furrow. Something must be in there stimulating the appearance of these nodes, which involves actin nucleation and myosin recruitment. The small GTPase rho turns out to be the key signaling link that stimulates the assembly of the contractile apparatus that drives the animal cell cleavage furrow. Now, when we talked about the first small GTPAs introduced in this course, RAN, I think I claimed that RAN was the first homolog discovered for RAS, the original small GTPAs. That might be wrong. I think Rho was discovered first, especially since Rho stands for RAS homolog. Now, RAN, the story is, it's a GTP binding protein. It hydrolyzes its GTP, but it does so slowly that it needs help from accessory proteins. That story is true for essentially all of the small GTPases, for Rho as well. One new wrinkle here, which is true for almost all of the small GTPases other than RAN, is that Rho, like RAS, is inserted into the plasma membrane by a lipid tail. I'm going to draw Rho schematically like this. It's a GTPase, so it's going to get a big capital G with a tail sticking down. And that's stuck in the plasma membrane. It's not that it's a transmembrane protein, it's just a lipid tail attached to the C-terminus of the protein. So this is true when Rho is bound to GTP. So this is Rho GTP, which is, in fact, the active form. So when Rho is bound to GDP, this lipid tail that anchors it in the plasma membrane is actually tucked inside. So I'm going to draw this as a cursive G instead. So that's Rho GDP. Again, exchange of GDP for a fresh GTP is greatly accelerated by an accessory protein in the context of cytokinesis. The relevant GEF, guanine nucleotide exchange factor, is a protein that was originally discovered as an oncogene, a cancer-causing gene that transforms epithelial cells. It doesn't matter really, but it got the name ECT2 from that association. ECT2 is the GEF for Rho during cytokinesis, which activates Rho by stimulating it to pick up a fresh GTP, which causes it to insert itself by its lipid tail in the plasma membrane. Now, that GTP-bound Rho does a few things, but one of the key ones is that it interacts with actin nucleation factors. We'll hear more about them later, to stimulate the production of long actin filaments right up there associated with the plasma membrane. So you can see where this is going. These nodes are going to have something to do with spots where active Rho appears. Ultimately, Rho hydrolyzes its GTP, but if you left it to its own devices, it would do so very slowly. As it happens, we think the gap for Rho in this context, which greatly accelerates hydrolysis of GTP and therefore eliminates active Rho from the plasma membrane, at least locally. We happen to think that the gap for Rho in this context is specifically attracted to long actin filaments. So again, perhaps we have a local source, ECT2, the Rho activator. We could localize it to the cell equator, where the cleavage furrow is supposed to form. We would get a high local concentration of active Rho. When we spoke about the RAN gradient around chromosomes, we had a distributed sink. The gap is everywhere, right? Everywhere in the cytoplasm, so that there's a gradient of Rho, a gradient of active RAN emanating from each chromosome. In this case, when we're talking about the activity of Rho during cytokinesis, the assembly of actin filaments here creates a delay in time, because it takes some time for a long enough actin filament to assemble, we think, to recruit this gap. Hence, we have a source that appears and is followed, if the GTPase is successful in stimulating filament assembly, is followed by an inactivation signal. So how does ECT2 get to the cell's equatorial cortex? I'm going to erase all this to describe the single most famous experiment in the history of studies of cytokinesis. Many of these experiments, including this one, start with a sand dollar egg somewhere on an island off the coast of Maine, and a fellow named Ray Rappaport. And Ray Rappaport, he's dead now, but he's famously quoted saying, "Never bet against the cell." Okay, so let's start with a sand dollar egg about to divide, as so many of these experiments do. And what he did in this one is he took a ball on the end of a stick, a pretty big glass bead, and shoved it down from the top all the way through the sand dollar egg to perforate it. So here's the glass ball, and of course it's on the end of a stick, which you have to imagine coming out of a chalkboard, and it deforms the sand dollar egg into a donut shape. Excuse me, let me fix that up a little bit. It's taking up a larger diameter on purpose, by the way, because of course it is getting squished into a donut shape, confining the mitotic apparatus to one side of this torus. So what do you think is going to happen based on what I've said so far? Well, what's the rule for animal cell cytokinesis? The cleavage furrow is going to cross the spindle midplane, right? So we know what's going to happen. It's going to cleave like that. The cleavage furrow should be induced in the patch of cell cortex closest to the spindle midplane. And that is, in fact, what takes place. No real surprise there. This yields a U-shaped cell with two nuclei. This is not the interesting part of the experiment. That's entirely predictable. The interesting part of the experiment is what happens next to this U-shaped cell. Well, it's going to go on into the next mitosis with two nuclei that are going to set up two cleavage spindles, two mitotic spindles, in each arm of the U. Now, of course, they're going to follow the rules, right? Cleavage furrow should be induced across the midplane of the mitotic apparatus, right? So we ought to get 2 cleavage furrows that pinch these things off of this remainder. The remarkable result in this experiment was that this U-shaped binucleate cell forms a cleavage furrow that divides the space between these 2 spindles as well. Hence, producing, if all goes well, producing in the end 4 uninucleate cells. We already know from experiments in which people destroy the mitotic apparatus with drugs or even take the mitotic apparatus out entirely with a needle that once the cleavage furrow is induced, you don't need the mitotic apparatus anymore. The cell surface will respond to it if it moves around in there, but you don't need it anymore for the cleavage furrow to continue. We also know from other physical experiments that the chromosomes are like have approximately the same role as the corpse at the funeral. They're the reason for the proceedings, perhaps, but they participate not at all. Okay, you can get a completely chromosome-free cell to cleave. Also, it turns out you can get a cell with 2 centrosomes, but no spindle fibers to cleave. From experiments like these, and especially this key one, the conclusion is, well, normally, the position where the chromosomes were and the midplane of the spindle, they coincide with the site between the 2 poles where all of these astromicrotubules from the 2 juxtaposed spindle poles overlap. And Rappaport and others said, well, there's something special about this condition of microtubule overlap, and these spindle poles here are also juxtaposed with each other, hence inducing the requisite geometrical condition here, even though there were never any chromosomes there and never any spindle fibers. So never bet against the cell. How does this recruit ACT2, however? I'm going to redraw that spindle in a little bit more detail. Here's my centrosomes again. I'm going to need orange for the ACT2 to highlight it, or red or something. So I'm just going to draw the microtubules in there in white. And I'm going to draw this thing at anaphase, when the chromosomes are separating, the sister chromatids are separating towards opposite poles, and there remain a few microtubules spanning the interpolar distance there. Let's not forget the astromicrotubules. They're growing out long now. This is anaphase. If you follow fluorescently tagged ACT2, then at just this stage, you start to see bright accumulations of ACT2 at the midplane of the spindle. Let me fix up these microtubules a bit. They're going to get longer as the cell goes further into anaphase and eventually into telophase. More of them are going to cross over. Of course, there's also, long microtubules out here and so on. As you keep watching, little spots of ACT2 start to show up here. Incidentally, this is this live imaging work was work that I did with my collaborators, among others. So it's as if ACT2 detects, ACT2, recall, is this activator of Rho. It detects some condition at the midplane of the mitotic apparatus during anaphase. These spots aren't there until the cell goes into anaphase. And then it's as if it climbs this ladder of astromicrotubule intersection points all the way out to where it eventually encounters the cell surface. And there it activates Rho, making that patch of cell surface recruit contractile proteins that then constrict the cleavage for Rho. The way this experiment here works is pretty straightforward, and we were able to visualize this by repeating Rappaport's experiments in the presence of marked versions of these essential factors, ACT2, Rho, and some of their friends, as well as being able to see where the microtubules went. So if I draw the juxtaposition between two spindle poles, I didn't draw them overlapping here, but ultimately they might. There's plenty of ACT2 and all its friends floating around in the cytoplasm. It turns out it doesn't have to recruit to the spindle first. ACT2 finds these kinds of junctions where anti-parallel microtubules overlap during anaphase. Hence, wherever two astromicrotubule arrays are juxtaposed, as they are here, there will be a little ACT2 accumulation that makes its way out to the cortex and stimulates Rho activation there, and the assembly of actin and myosin to constrict the cell surface.